Okay, so in this video, we're just going to talk about a little bit about the mitochondria and about the electron transport chain in general. So the mitochondria is an organelle that we see in aerobic organisms, and it's going to be involved in oxidative phosphorylation in the electron transport chain. So just to briefly go over the structure of a mitochondria, we have an outer membrane, which you could just refer to as the outer mitochondrial membrane. We have an inner membrane, which is really where we're going to be talking about all of our processes happening. And we have two compartments of our inner membrane, and that is the cristae, where all of our electron transport chain proteins are going to be embedded in these invaginated folds of the inner membrane. And then we have the inner boundary membrane. So all you really know about the inner boundary membrane is it's located in between these cristae, and it's responsible for the transport of proteins into or through the inner membrane of the mitochondria. So another important thing we need to remember are the two compartments of our mitochondria. So the very inside of our mitochondria, inside the inner membrane, we refer to as the matrix. And the space between our inner membrane and outer membrane we refer to as the intermembrane space. Okay, so just a quick overview of the metabolic processes we've talked about so far. For the last test, you had to study the mechanisms of glycolysis. So that happens outside of our mitochondria. For this exam, we talked about the TCA cycle, which is, you call it TCA cycle, the citric acid cycle, the Krebs cycle. So that happens inside of our matrix in our mitochondria. And then for this video, we're going to be talking about the electron transport chain, which happens, these electron transport chain proteins are actually embedded into the cristae of the inner membrane. So a good rule of thumb to remember for a lot of your mechanisms is you're typically going to be making energy-carrying molecules or energy-carrying cofactors like NADH, FADH2, or NADPH. And if you don't remember, NADPH is just like NADH, except it's carrying a phosphate group. It carries a lot more energy used to make a lot larger molecules. So like I said, a good rule of thumb is to, wherever we make our energy-carrying cofactors or energy-carrying molecules, we're typically going to make them on the same side of a membrane as where they're going to be used. So we're going to be making these energy-carrying molecules in the matrix, and they're going to be sent directly to the inner membrane at the electron transport chain proteins in those cristae. So one thing to talk about really quick before we start talking about the electron transport chain is the concept of redox potential. So redox potential can easily be translated to oxidizing potential. And if you remember what it means to oxidize something, it means to take electrons away from something. So oxidizing to take electrons away from something, and then we have reducing something, which is to give electrons to something. So when we're making energy-carrying molecules in the TCA cycle, we are oxidizing whatever molecule we're talking about, be it pyruvate, oxaloacetate. We're oxidizing that thing and giving those electrons to an energy-carrying molecule like NADH or FADH2. So in the TCA cycle, we are oxidizing the forms of glucose and reducing these energy-carrying cofactors. But in the electron transport chain, we're going to be talking about it a little bit differently. So in the electron transport chain, we start at a point of really low redox potential. And what that means is we start at a point of really low desire for electrons. So when we start our first step of the electron transport chain, be it for NADH or FADH2, the first carrier that we give it to is going to have a really low redox potential, meaning it has a really low desire to oxidize NADH or FADH2. And what that can be translated to is it has a really low desire for electrons. And that's really important when we're talking about electrons traveling through the electron transport chain, because each carrier that we pass on our electrons to through the electron transport chain, in whatever path we're doing it, in the NADH path or the FADH2 path, we're going to have an increase in redox potential. So what this means is the molecule that we are handing off electrons to, say we're handing off electrons from this green circle to this red circle, that means that this red circle is going to have a more desire for these electrons. So the reason these electrons are being passed on throughout the electron transport chain is because the next cofactor has a greater desire to obtain those electrons. And that's just something that's something that's important to note. Remember the last electron acceptor, the thing with the highest redox potential in the whole electron transport chain is oxygen, O2, and we'll talk about that in a second. Okay, so let's go to the structure of the electron transport chain. So what we're looking at here are the four complexes of the electron transport chain. We have complex 1, 2, 3, and 4. So we're looking at these electron transport chain proteins embedded into the inner membrane. So the outside here at the top will be the intermembrane space. We'll just write inter. And the inside will be the matrix. And the whole real point of the electron transport chain is to use the energy that's being stored in those electrons to cause a pumping of protons into the inner membrane space from the matrix. So we want to make some sort of proton gradient inside the inner membrane space to the matrix. That being a higher concentration of protons in the intramembrane space. Okay, so one thing to note is there are two separate pathways that we could take in our electron transport chain. The first pathway will be the NADH pathway. So NADH. NADH will be oxidized to NAD plus and H plus. And the two electrons that are being stored in NADH will be passed on into the complex 1 of our electron transport chain. So conveniently that complex is called NADH dehydrogenase. And you will need to know all the names of the carriers and complexes for the electron transport chain. And like I mentioned earlier, all of our energy carrying cofactors, be it NADH, FADH2, NADPH, they're all going to be holding 2 electrons for what we're talking about. So just keep that in mind when we spin the molecule of NADH, we spin the molecule of FADH2, that we're donating 2 electrons. All right, so we oxidize NADH to NAD plus and H plus, and we are going to pass on our electrons to an electron carrier called FMN. So FMN is one of our flavoproteins that we discussed in lecture. You just need to know what its definition is, and you need to know that FMN is the first electron carrier in complex 1. So we are going to be giving our electrons from NADH to FMN. Okay, so FMN is going to hand off our electrons into an electron carrier, which we'll talk about in a second. But as electrons travel through complex 1, it's going to be pumping out 4 protons. So as every pair of electrons travel through complex 1, 4 protons will be pumped out from the matrix to the intramembrane space. Okay, so the electron carrier that we're going to be talking about is called ubiquinone. So an important thing to remember about ubiquinone, and we'll talk about a similar molecule in the light-dependent reactions in photosynthesis called plastoubiquinone, is when ubiquinone is the oxidized version of this carrier molecule, but the reduced version is called ubiquinol. So just remember the oxidized version, ubiquinone, reduced version, ubiquinol. So from FMN, we will be going through some intermediates which we don't really talk about in class, and we'll be handing off our pair of electrons to ubiquinone, which will then reduce it to ubiquinol. So I will just do the path of electrons in gray, if you could see that. So this electron carrier will carry our electrons from complex 1 to our complex 3 right here. So it goes from complex 1 to complex 3 via ubiquinol carrying these electrons. So another pathway we could take is through complex 2. So complex 2 is right here. So this complex is called succinate dehydrogenase. So this enzyme is the exact same enzyme that's number 17 in your TCA cycle. So this enzyme is actually coupled to the electron transport chain. This enzyme is actually embedded in the electron transport chain. So it's the same exact enzyme doing the same thing. So when you learn TCA cycle, you probably memorized that it makes two molecules of FADH2. Well, you can think of it that way, or you could also think of it as directly handing off two electrons to succinate dehydrogenase in the electron transport chain. Whatever helps you remember exactly how it works. Also remembering that this enzyme is both in the TCA cycle and the electron transport chain could help you remember that the TCA cycle happens in the matrix, because it would have to directly hand off, or it would have to directly be involved in this inner membrane for the succinate dehydrogenase. So in this reaction, we have succinate being oxidized to fumarate. In the TCA cycle, if you were to think of it like FADH2, you would think of it as FADH2 being oxidized. But I think for the electron transport chain, you need to know that it's succinate being oxidized to fumarate. So it's handing off, succinate is being oxidized to fumarate and handing off two electrons to our succinate dehydrogenase. And our first electron acceptor in succinate dehydrogenase is FAD. So an important thing to note about complex 2 is the energy that's being released by this oxidation of succinate is not strong enough to pump out protons into the intermembrane space. So your complex 2 is going to be the only one that does not pump out any protons. Okay, so succinate dehydrogenase will pass off our electrons to another electron carrier to complex 3. And this electron carrier is the exact same electron carrier as what's used to transfer complex 1 electrons to complex 3. So this is ubiquinol. Ubiquinone when it's oxidized, ubiquinol when it's reduced. So from here, either electrons from our complex 1 will go to complex 3, or our electrons from complex 2 will go to complex 3, but it'll have the same end result. So the name for our complex 3 is cytochrome BC1. Very similar to our cytochrome B6F in the light-dependent reactions of photosynthesis. So cytochrome BC1 will catalyze a transfer of electrons from our ubiquinol carrying molecule to another carrying molecule called cytochrome C. So cytochrome C. So just like in complex 1, with each pair of electrons that come through our cytochrome BC1, there will be a pumping of protons from the matrix to the intermembrane space. So just like in complex 1, every pair of protons will be pumped, or every pair of electrons will be pumping out 4 protons into the intermembrane space. So if we were to take account right now of how many protons are being pumped out for one NADH molecule, we'd have 8. And if we were to count how many that were being pumped out for every FADH2 molecule at this point, we'd have 4. Or every succinate molecule being oxidized, if that's a better way to think about it. So our cytochrome C will carry these electrons to our fourth and final complex called cytochrome C oxidase. So that's pretty easy to remember. It's responsible for oxidizing cytochrome C. So cytochrome C oxidase is going to, oxidize cytochrome C and catalyze there's a transfer of electrons from our cytochrome C to our final electron acceptor with the most redox potential of this pathway, and that is oxygen. So as every pair of electrons that come through cytochrome C oxidase, it's going to be responsible for reducing an oxygen molecule and reducing an oxygen molecule and creating one water molecule. So this is going to take two additional protons from the matrix and use those two electrons to form the bonds to make a water molecule. So the energy released from this reduction of oxygen is going to cause a pumping of two protons for cytochrome C oxidase. This will be our final proton count. So for every molecule of NADH that is oxidized at complex 1, we'll be pumping out 10 protons. And for every molecule of FADH2 or succinate that is being oxidized at complex 2, we'll be pumping out a total of 6 protons. Right, and this whole use of oxygen and these differing redox potential molecules to cause electrons to be carried through the electron transport chain, we could refer to that as oxidative phosphorylation. So that's oxygen being, the reason that is because we're going to use this proton gradient to make ATP. So we're going to be phosphorylizing ADP to make ATP, and the whole thing is caused by this redox potential gradient, which eventually would be caused by our oxygen molecule at the very end with the most redox potential. So we are creating this proton electrochemical gradient that we will talk about in the next video. And not only are we creating this electrochemical gradient, which we could refer to as proton motive force, but we're also creating a pH gradient. So the intermembrane space will have more protons, a higher concentration of protons, which will mean it'll have a lower pH. Right, because we could think of proton concentration as being inversely related with pH. And the matrix will now have a higher pH because it's got less protons in the matrix because we pumped them all out to the intermembrane space and we've taken up 2 more in the creation of water. Okay, and one last final thing to note is remember that from the beginning of a pathway, be it where NADH starts at complex 1 or where our electrons from succinate start at complex 2, we are going to be going in an increase of redox potential for everything that's handed off. So our ubiquinone would have a higher redox potential than our FMN or our FAD. And also, we have a decrease in energy being provided by the electrons. So as the electrons travel down the electron transport chain, they're going to be losing energy until we get the right energy state, which is perfect for reducing oxygen to form water molecules. So just remember that higher redox potential as we go, lower electron energy. Okay, so this is just a brief overview of the electron transport chain. There's a lot of other definitions. You need to know a few definitions for your cofactors. You need to know, I would just memorize everything in the PowerPoint and know for sure that you know it well. And next videos we'll talk about photosynthesis and ATP synthase. So ATP synthase using this proton gradient we've just created to catalyze the reaction of ATP formation.